All right, so this is where you still see this screen and hear me well? Okay. So far I have talked about static shapes, individual shapes, and to sort of quantify shape differences. That's so far what we have done. We have done it for points, and we have done this for curves, and we have done this for surfaces. And very quickly I showed you we have done this for branching structure, tree-like structures, and graphs. But now I want to talk a little bit about shapes that change in time. How do we mathematically consider them, and how do we model them? And so we are going to think about shapes changing over time as a continuous process in some kind of a shape manifold. As we know, it's a nonlinear space, the metrics, the quotient spaces, this and that. And so in this picture, you see there is a shape that is evolving over time, and each time it's a point in a shape space. So each point is a shape, and somehow it's a continuous trajectory, right? So what are the challenges in this sort of viewpoint of trying to understand the dynamics of the shape? Firstly, of course, shape is a nonlinear manifold, so that's that. There's high dimensionality, both spatial and temporal. The shapes could be high dimensional. We have surfaces, and these are continuous surfaces. And then, of course, in time, if you have many measurements over time, this sort of adds up. So thinking the whole trajectory, it's a very high dimensional data object. Then, of course, the question that Maha brought up and is high on our minds also is that if you have missing shapes, noisy shapes, broken shapes, and all the imperfections, so we are developing the mathematics in some sense for very pristine data, but then real data has its own problems. One more issue that I want to bring up is this synchronization. So you are observing some tree, maybe a biological growth, and I'm going to show you an example of a small seedling growing into a tree. And if you follow multiple such growths, they grow at their own time rate. They are not sort of synchronized across times. And in trying to compare these things, you have to reconciliate between their different evolutionary rates. And so that's also an issue, and we'll handle that, and many more. And so a few applications that we have looked at, one is how to model shape a skeleton of a human worker in an industrial setting, performing in front of a workbench or something, like IKEA type model. They are following a recipe for assembling a product, but long time sequences. And the goal was how to simulate new sequences, such thing, because it's expensive to collect the data. We have a collaboration with Samsung, and they gave us few sequences, and they want us to learn model and generate many more new sequences, which essentially requires understanding the underlying model. We have data for cell motility, the cells move, and they have kinematic side of it, but they also have shape dynamics, how the shape moves. Cancerous cells become more motile, they are freer to move, and there are changes in the shape that makes it easier for them to move. And so we are interested in sort of shape evolution. And then this last point I'm going to explain a little bit in an example of what is, if you focus on some sort of anatomical structure, in this case, this is hippocampus. What is an average human shape evolution from an age group, in this case was 22 to 90? Just some anatomical part. You have observations from different people, and can we use all the information pulled up and to talk about average nominal growth? Hi, Maha.
Since in biology, often growth is critical, therefore this Kendall description of translation and rotation is correct, but scale is no longer correct.
Yes.
So, are you going to address that at all?
Yes. I mean, that's rather easy. So we just, at some stage, we removed scale. We just don't do that part, and everything else is fine.
No, but the problem is that scale is not going to be uniformly you cannot remove it uniformly. Growth is often non-uniform.
Yeah, so that's why we have this sort of, where did it even go? We have sort of non-linear registration techniques from time t to time t plus 1. We have just think about the main branch of the tree. We have non-regist. What we are not going to do is just skip this step of this rescale the metric. We'll keep the original scale. And then we are going to do the registration using this elastic metric, and it may tell us that one part elongated more than the other part.
So,
And that,
so,
yeah.
so you're saying that mathematically, are you saying that the trace of the first fundamental form is something which is going to be spatially and temporally inhomogeneous?
Yes, that's part of our
Yeah.
descriptor,
Okay.
right? I have this G comma N matrix. I keep the G and I keep the N. At each point on the surface, I have a G and
Thank
I
you.
have an N. And I'm comparing across. And so if I'm doing it across two times, for one object to another, these can change.
And is there a way of introducing a micro scale? Because right now your descriptor ignores the fact that, for example, biological tissue is made up of individual cells. And so can you build in the fact that certain cells are dividing and certain other cells are not dividing, or you essentially have smoothed out over that and so you've lost that information?
We haven't actually worked with that specific problem for that reason. Is that cell grows and then divides, and
Yeah,
for us it becomes a topologically complicated object.
exactly. So you
And
just smooth that out.
yeah, we just outer contour or something,
Yeah.
as long. But that's, yeah, this is one of our ongoing NSF, which we have one component is sort of dividing, meiosis, mitosis in
Thank
the
you.
in the setup. I just want to mention in this context that one sort of pre-processing step, which is when we get objects, we get them at sparse times. Of course, I'm talking about as a stochastic process which has continuous time. And so we are thinking of this as continuous trajectory, but we have only sparse time data, sometimes very sparse data, but sometimes just sparse data. So we have these tools which are adapted to the geometries of the shape manifolds, things like splines. So we can take these sort of discretely, timely observed objects and interpolate between them using these spine type tools and build as dense sort of continuous evolution as you want. We can sample them. And in this case, I mean, I have one example. This is a toy example, but I want to show you this practical example that we wanted to build a continuum of a trajectory of this hippocampus in human brain going from age 22 to age 90. But as you know, we don't have the data for an individual. We have, in fact, very sparse, at most maybe 2 or 3, 4 scans per person. You don't have too many scans, and these are usually limited. And so we use these statistical tools, which we can apply to all the other kinds of data too. We pull data from 3 different data sets, HCP, OpenPin, and Indigo. One data set was for younger people, other was for middle-aged, and other was for older people. We pooled the commonality of structures across humans and kept the individuality. And in statistics, there is a name for it. These are called mixed effect models. You have sort of an overall mean, but you have terms for the individualities. So I'm going to describe a little bit more about the tools that were needed to go into this. But the bottom line is at the end of it, we were able to estimate in a statistical sense what is an average trajectory with data that is coming from a few scans per individual, but lots of individuals, tens of thousands of individuals.
And the smoothness descriptor is some what
It's
norm do you have to use? I missed that. Sorry.
smoothness in time or smoothness in shape.
Both, both, both.
So smoothness comes from the sort of the basis we have chosen for the sort of temporal representation.
Are there
It's a
optimal bases?
no, they are optimal in the PCA sense.
But PCA is linear. So is
Yeah,
that
so we do
problematic?
linearize. Yeah, we do linearize these into these shape spaces. They're sort of more into in the second part where we do the spatial PCA, shape PCA, go into the tangent space, and so then they are linearized, respecting the geometry of the shape space. And in that space, there are this sort of well-known statistical problem about curve fitting using sparse measurements. So then we can deploy linear tools, right? And
Are
so
there are there priors associated, for example, with geometric flows There are all
We
these
did not.
Monge Ampere or Ricci or Yamabe and all these kinds of mean curvature flows and things of that sort that you might be able to
We
deploy
can.
in.
Yes. It can be deployed as a cost function in our optimization steps.
And the reason I ask is I think Keenan is going to probably talk about some of these things in his tutorial on Thursday, and I wonder whether there is some common
Yeah,
ground.
right. So when we do functional data analysis after all this linearization, just functions in time, we have some sort of standard metrics. But that can be done with some non-standard metrics also. One can introduce penalties and other incentives to select the bases accordingly, right?
Thank you.
So once it's linearized, I think we are sort of open to many
Yeah.
in okay. So now I'm actually going to switch my talks. This is what sorry, I'll have to go all the way back up. This is where I wanted to do, and I'm about 10 minutes late into this. That's fine. This is a shorter talk. Maha mentioned some of the references. So we have a couple of textbooks for curves and functions. There is an ex-student of mine who maintains a very good database, sort of code in Python, R, and MATLAB, and so on and so forth. It's very updated. Thanks to him. I'm not good in this stuff. For 3D objects, we have some sort of scattered code we can provide. Certainly, it exists. But what I'm going to talk about a little later about dynamical shape, that's our new ongoing project. We have some codes, but not as polished and ready to share. But surely we can pass on things with enough asterisks attached. So this is where I am. I want to start talking about statistics. So I will talk about static shapes, single shapes, how to compute shape summaries, put statistical models, compare populations. You have population with treatment, population without treatment, how to do the significance test between shape populations, and then finally how to model these dynamical shapes. All right, so maybe this can be skipped. I've sort of mentioned the goals that I want to have. These are a little bit more detailed. So I'm going to start with the simplest notion. What is a representative shape? And one can use the notion of mean or a median, and people are talking even about the mode in a shape space. So it's rather simple for us. We already have defined a shape metric, which was the closest distance between the two orbits. So once we have a shape metric, we can define the mean shape or sample mean as a shape that minimizes sum of squares of these shape distances to the individual shapes. It's a very common notion that has been used and analyzed for many different kinds of manifolds over the years. There is a neat algorithm. Actually, sorry, I should have put it full screen. There is an algorithm that goes with it. Now that I put it on full screen, I can't do what I was trying to do. Let me take it back. There's an algorithm which is an iterative, which is you have a current estimate of the mean, you align and register the individual shapes in their orbits to the current mean, and then you update the mean. And you keep going back and forth. And people have studied how good is this algorithm, local convergence, global convergence, and so on. I'll just show you some examples. This is the example for just scalar functions. So I have some unimodal functions. It's just a toy data. And I use this algorithm to align an average. And so if I don't align just simply average, I get this black curve. But if I align and then average, I get this black curve, which is a better representative of the shape because these were unimodal curves. This is real data. This is some sort of a COVID data over a 1.5 year period, 25 European countries, the peaks and valleys, the arrival of these different viruses, versions of viruses. And the question was, is there a trend to it? Is there a pattern to it? And if we just average these infection rates without any sort of shape consideration, you get these black curve. But if you align these curve and then you average them, then you have a very prominent description of the peaks and valleys and some commonality of the structure.
Can I ask a quick question, Anuj? So,
Yes,
is there
yes,
dynamic time warping in here by
yes.
align? Yeah, and
Yeah, so it
maybe
is
I missed
dynamic
the
time
detail.
warping in the tool is that the cost function that has been traditionally used for dynamic time warping is not the one we are using. But dynamic time warping involves optimization over time warping. That part we are doing, but with our cost function. As I mentioned in the previous talk, the dynamic time warping traditionally uses L2 norm, which is degenerate. So how they handle is they start putting some sort of penalties to not do too much warping. But our metric has, of course, I'm gung ho about it. It is our metric. It has a lot of nice mathematical properties, doesn't require any penalty, and these are the results. In fact, the only complaint I've heard about this algorithm over the last 15 years is it does a too good a job of alignment. If you notice, it sort of even aligns the smaller peaks, which might be noise. So it starts aligning noise, and then it ends up looking like some interesting structure, even though it was noise. So
So,
that's, yeah. Go
so can I
ahead.
ask a question in this context? What do people do when you have very intermittent signals? So, for example, I'm speaking to you, and there's a lot of gaps, and those gaps
Yeah.
are not terribly important. I mean, they need to be there, but the length of the gaps is not important. And why do I say that? Because I can essentially listen to a podcast of this conversation at 2x, and I can still understand pretty much everything. That means that there is ability to there should be an ability to compress without losing meaning, because you're not essentially changing the frequencies, you're only removing the gaps. If you were to do a uniform compression, then all my speech will essentially go up in pitch, but it doesn't. So what do people who use dynamic time warping in audio use relative to what you are suggesting? Question one. And a related question is, almost certainly what they're doing is removing the gaps, which means that they are very strongly non-linearly doing this time warping. And so I want to come back to the question from before, which is, instead of doing an L2 with respect to the square root velocity norm, can you do something different for certain kinds of time series which are strongly intermittent, like speech?
So my suspicion is we can do, I mean, I'm not very confident what I'm saying, but we can do sort of removal of gaps in the same way as they are doing. Because if there is a gap, it's a gap in whether it's in the original signal space or this transformed square root space. A gap is a gap, right? So we can still remove the gap in the same L2 norm as they are doing. Those
But
tools
you
are
don't
still.
want to change the pitch. So if I do dynamic time warping in your using your approach, my pitch will change.
Yes, yes. I should clarify that. I don't quite do, so I'm still doing time warping in the original space. There is a corresponding group action in the square root space, which is a different group action, and that group action commutes, right? So, I wish somewhere in the previous slide, maybe I stopped. It's a different group action. I'm not doing time warping in the square root space. I'm doing is like the corresponding group action. So it's always as if I'm time warping in the signal space. But the group action is different, and
I
it's the
see.
compatible one. And let me see. There is a square root of gamma dot that comes out.
Yeah, that's a Jacobian.
Exactly, that's the
Yeah.
Jacobian, and that is maintained for 3D objects, for curves. This you will see it. Where is it? Have I even written down this? I haven't. I sort of worked around it somewhere. Sorry, I don't have
No,
that.
it's okay. It's okay.
Yeah, but there is actually, there is a corresponding group action. So it's not as if I'm time warping in this transform space. Whatever the group action I'm doing in this space is equivalent to time warping in the original space. So I think I try to answer the second part of your question, which is gaps. We can try to do the same thing as they are doing, and not lose things. Same tools can be applied,
Thank you.
right? The first question I wasn't quite sure, so I'm just going to ignore it. These are difficult questions to answer. Here is an example of averaging shapes. So just like you average functions, you can average curves in terms of their shapes. And if I take these original objects and just randomly rotate them, translate them, scale them, the point is this answer will not change. This analysis is invariant to these things. That's the way the mathematics has been built in. We have done this for 3D objects. We can compute average shapes in that sense. And these shapes are invariant to reparameterization of the surfaces. You can give me surfaces in arbitrary meshing. I will remesh it, reparameterize it in the optimal sense under my metric. So it will be invariant in that sense. How does one do beyond the mean? That's the first moment, but the second moment, covariance. Covariance is a linear operator, so we cannot directly define covariance on the nonlinear manifold, but we go to the tangent space at the mean. We select the mean, we have a tangent space at the mean, we map all the given shapes to these shooting vectors, and that's where we compute the means. And so here is an example. Again, these sort of made up shapes. All of these are circular objects with two handles. Just the handles are close or further apart. That's the difference, right, in all of these objects. So it's a made up to make a point, the simulated data. If I just do the averaging without any registration, just thinking of arc length parametrized curves. Suppose each one of the curve is arc length parameterized, I'll get an average shape, which is this, because these handles didn't line up. They got smoothed out. And the PCA directions look like I'm killing or birthing these handles, right? On the other hand, if I do it with registration, I do the nonlinear registration along the curves, these handles will match across objects, and when I average them, I preserve the handles. And this is what I love. This is beautiful. That the principal direction actually corresponds to stretching and bringing these handles closer and further apart. So this is a phenomena. We just introduced this in the data, but we didn't tell this to the algorithm. But here, not only the average has both the handles, that's fine, that's part one, but the most dominant direction is just moving the handles closer and further apart, which is how we generated the data. And then we have other principal directions. Yes?
Can I just unpack this a little bit? In your elastic shape analysis, are you allowing for constraints on the enclosed area or only looking at the curve?
Only at the curves.
Okay, so that means that, again, it becomes only a function of the velocity along the curve and then the tangent variation.
Yes.
So
yes.
It's really bending and stretching in physics language.
Yes.
But okay, thank you. If you wanted to allow, because if I'm thinking about what a pathologist does, or a cell biologist does, would be to essentially say, oh, I'm looking to try and understand this difference in the shape of cells. So it's a 2D projection of a three-dimensional object. Then there may be an additional constraint associated with the integral of r d theta, which is in this case the area.
Yes. One can do this in several ways. One is you can extract, which is what we did with cells coming down in this talk. When we work with cell motility, we just remove the areas from each one of them and separate them as a separate scalar function. We model it by itself. Or leave it in there, but don't rescale the curves. So whatever the area enclosed, it remains there. You don't change.
And that means you'll have to weight how much you're penalizing area changes compared to arc length changes compared to curvature. And that's now a choice that you have to essentially figure out somehow. Somebody from outside has to tell you that.
Right. You can choose weights if you want to do it. I mean, it depends on how you reincorporate that information. There are several choices there. But since it's a scalar one, area is a scalar thing, it's not that much more difficult to incorporate.
Thank you.
Yeah. Okay.
Vanessa, I think, has a question.
Yeah, I was thinking about something. This is very cool. And you have an example here where the shapes are sort of really covering much of the space that is available to move these two handles. How would this analysis look like if you have something like there are 2 or 3 states that are most often reached and you don't see anything in between? Is that something?
Good question. So what we do is we sort of bring in clustering as an intermediate step. So we have shape metric, we have clustering tools, we have some sort of good mode finding methods. We'll cluster these shapes into their groups, and within the group we will do this.
Oh, okay.
Right. And then if you want to go across the clusters, we have geodesics, which will take you and you can observe and study how the mean of this cluster changes into this. You will see a third handle growing, you know,
Mm-hmm. Mm-hmm.
and that sort of thing, or a handle being killed and all that. But within the group, the handles will just simply be moving apart. Okay. What did I do? Okay. So we did this with some real data, and the differences are somewhat subtle, but very interesting. But I'm going to skip this because time is short. I do want to get to the dynamic shapes, I think, which is in use. This I want to mention because it was just a fun thing, and it's one of those things you are very excited about something and nobody else is. So anyway, I throw this in my talk. So we developed this technique for estimating shapes of chromosomes from the single cell Hi-C data. That's fine. So we had lots of chromosomes. These chromosomes have some kind of segmentation. You can chop these chromosomes into smaller pieces using this notion called TAD, which somebody gives to us. What we noticed is that these smaller pieces, these curvelets, have very few shapes, distinct shapes. They all were of certain types. So it's like proteins. Proteins are a sequence of amino acids. I thought, why can't we come up with just some names for these little pieces? And a chromosome is just a repeated use of the similar shapes put together. And so we call these shape letters, and we built a shape alphabet. So we would chop curves, big curves, hundreds of curves, thousands of curves, into these smaller pieces, and we'll cluster them into similar shapes. And we'll take the mean or medians of these clusters, and we'll call them these shape letters, shape alphabets. And then once we have these alphabets defined, we can take individual chromosomes and we can just write them. I mean, we needed 45 or so we went capital and small. We ran out of alphabet. And so you can take a big compound like that, and you can chop it into these sort of shape letters and build express curves as these letter sequences, and you can compare them. So for example, from the same data, if you have a random algorithm which is generating sort of slightly different shapes every time you run the code, and you want to compare them, you can compare them with these sort of representatives, which I thought was rather interesting, but nobody seemed to bought it every time we presented it. Anyway, moving on. So we can do this with sort of mean and PCA with three-dimensional shapes. And then trees. This was very difficult. I mean, it's even mind-boggling to ask a question where you have trees from different species, and we are hell-bent on averaging them just because we have a program to do it, whether it makes sense or not. And so the average tree represents like what are the common structures, and if the trees are very different, there is not much common to it, right? But we can do it. But it made more sense within the species. So within the species, we have lots of trees, we had samples, we could compute their average. We could study the principal directions variability, the principal mode of variability. So in the middle, you have the mean. Going left is the negative first principal direction. Going right is the positive first principal direction. Each row represents a principal direction: first, second, and third. And so with very few principal components, you can capture this high dimensional variability, which would have been mind-boggling. Like, how do you even handle the variability across such trees? But with about 10, 20 principal components, we got it, we nailed it. In fact, we generated random Gaussian trees by putting these models on these principal coefficients, principal scores, and here are the kind of trees we get. Okay? One can debate it good or bad, and here are some more examples from other species. But in other words, we have now this ability for trees and 3D objects or curves to be able to talk about mean or clustered means and PCAs, and impose some statistical models on principal coefficients. Principal coefficients is a way to reduce the dimension from a very complex tree structure to just, let's say, 15 to 20 principal coefficients in the tangent space of the tree space. And we have done this with graphs. These were some sort of blood vessels, somewhat simulated. but rather complex problem. This was mind-boggling when we was trying to start it. Like, how do you even quantify this? But nowadays we are talking about using these structures to detect glaucoma, and some reasonable success. Not too exciting, but so some kind of case studies that went along with this. So first example, here we were studying mitochondria in the muscle tissue in mice. We had two populations. One was mice which were sedentary, which were not doing much, and other who were made to exercise for food. So two population of mice, exercising and not exercising. And the question this biologist asked was, is there a statistically significant difference in the shapes of their mitochondria in the muscle tissue? So whether it's a good question or bad question, I'll leave it up to him, but that's the question he posed to us. In fact, he gave us data where he hand segmented. This was before he became famous. He was young, he had plenty of time on his hand. He hand segmented these mitochondria in the stained images, muscle tissue, microscopic images. And he gave us two kinds of mitochondria. One he called SSS, one is called IMF. So here is SS and IMF, and you can see they are very different in shapes. But this is SS of a mice which was exercising, and on the right is the SS mitochondria of a mice that was not exercising. And this was not at all, you can't eyeball it. It was a population question. It was not a question about individual shapes. And so the goal was how to compare the shapes. We derived this statistical two-sample test. And when we compare the SS with IMF, they are very different shapes. So no wonder the test statistics is way out there. These are very different shapes. But in this SS versus IMF, where they were not so different, visually not much different, there was statistically significant difference. And this difference you could not have found using Kendall's method or non-elastic method. The elastic method, which additionally does registration of points along the curve, is able to remove some extra nuisance variability and then be able to tease out the differences in the population. So I think Salem asked the question about metrics and choices of metrics and which one is more appropriate. And I think this was one of an example. In this paper, we actually studied 5 or 10 different metrics to show with the same pipeline, but different metrics, and see which one can distinguish between populations. And elastic metric was successful. And we have done this sort of pipeline for lots of medical problems. So in this case, let me just move my screen to see what is the, we were studying brain anatomical structure, and the goal was, to study the PTSD disease. Can you use the structures of subcortical structures, shapes of those, morphologies of those? And so we had a pipeline where we'll start with original data, segment them, compute the means, compute principal components, and use these principal component scores as shape features to feed into some regression models and try to predict the clinical outcomes for PTSD. And I mentioned these shape features. These shape features are invertible in the sense that you can take these principal components and map all the way back, and you can reconstruct individual objects. And we were able to visualize along what direction that prominently separates the healthy versus controls. So help doctor visualize what deformations of these structures are the most distinguishing directions. So now recently I'm working with the spine and vertebra and morphologies of spine to predict a number of diseases. And this is very interesting because even according to the doctors, a lot of these diseases is about morphologies, the shapes of vertebra and their relative alignment with respect to each other. They have names for these individual diseases. So this is very appropriate for shape analysis. And we are beginning to work on doing shape PCA to extract essential variability and feed some regression into regression models. So these vertebra would go in and we'll try to predict the gradings. Okay. There is one more couple of slides about actual regression models, but that's not so important. I think we maybe from a statistics point of view it might be important, but not. I want to get to these dynamical shapes, and my pitch is learning morphogenesis from data. If you have plenty of data, this is not at all to replace the sort of the biomechanical models or biological models, but to complement them. And so this is what we want to do. If you have lots of data, biological growth data for individuals, you can view them as trajectories or stochastic processes on a shape manifold. And can we learn the underlying averages and variations, not in individual shapes, but how they are growing or changing over time. And so we have divided this sort of into space and time. Sometimes you have discrete shapes like we had skeleton, or you have a continuous or dense shape like surfaces. Sometimes you have discrete times. Sometimes you have continuous time. So you can break this up into these different arenas. And so this is a slide that I showed earlier, and I want to now talk about how do we handle these issues. So the first thing is nonlinearity of the shape manifolds. So I did mention this in answer to a question earlier, that we have this way of flattening the manifold locally, either with one single tangent space or a collection of tangent spaces as needed, and make these shape features into Euclidean vectors, and then apply more classical statistical techniques. There was this issue about synchronization. So I mentioned a project where we have a human worker performing a certain activities on a workbench. Every time the same worker performs these activities, the rate of evolution is slightly different, right? 5 minutes into the activity, the person may be further along or less further along every time he does it. And so one of the things we have to worry, we call it the phase variability. Just like I aligned scalar functions earlier, I showed you COVID data. In the same way, we align shape sequences. And we have tools for that. Again, very, very solid tools, not much human intervention. And here is an example. On the left are shape sequences, person performing activities that are not aligned. On the right, we have after alignment. So we have these sort of so-called 4D sequences, 3D plus time, but for individuals. And we have to synchronize them before we start averaging across sequences. Go ahead, Noah.
Can you comment on the necessity or utility of landmarks here? So I'm thinking that if you just had meshes which were unstructured versus having meshes with identified nodes and the joints of the skeleton or something like that.
Yeah, so there are two different things going on. One is the individual shapes have to be spatially registered, like which point on this shape matches which point. The thing that I'm talking about is temporally registered, which time in this sequence. And so in none of those two processes I assumed any landmarks. Now, if you have landmarks which are trusted, then we have ways of utilizing them. But I did not assume anything.
So in this, perhaps it's a cartoon. Oh, okay. So in this case, right, are these mesh triangulations?
Yeah, these are triangulated meshes. We treat them as parametrized surfaces, so we sort of do something slightly different.
But are they parametrized? I guess maybe to the core of my question, are they parametrized such that there's a vertex at the hinge?
No.
No. Okay. So every time you have a sort of a new remeshed surface.
In fact, you can give me an arbitrary mesh and we will remesh them or reparameterize them to spatially register them across each other. Yeah. So there's no landmark involved on them in space, on shapes.
Okay.
And there's no landmark involved time-wise either, because that would be neat if there was if you could identify that raised hand should be matched with raised hand. But that's an outcome we want rather than imposing.
Right, of course. Yeah, absolutely.
And yeah.
Anuj, can I ask, this is a follow-up question, and it's very interesting because in development, we always have this problem, right, of the timing of
Yes.
embryos that do the same thing, but they might develop a slightly different rate,
Yeah.
and but the differences are kind of subtle, right? So in this example that you show of the people jumping, the movement is dramatic with respect to the shape of the overall mesh that you have.
Yes.
Do you have a sense of how small changes of a mesh can you use to do this sort of
Yeah. So
registration?
at this stage, you give us data, we apply it. The question is what is detectable and what is
Mm.
missed. That remains to be seen. I think I'm going to show you more examples, and maybe some more examples will help. And this is real data, especially of these plants growing. These are very subtle changes and completely misregistered. Plants grow at their own rate, and sometimes we even have a lot of missing data. Um, I don't think I have a good answer to your question in terms of quantifying how subtle the changes could be.
Mm-hmm.
But the spirit is to align them as well as you can. And as I showed you with the scalar functions where we were aligning peaks and valleys, this method has only been criticized that it is too good in aligning. That's the only in last 15 years. Um, as compared to DTW's dynamic time warping of many different kinds. But remains to be seen, I think, case by case basis. So this was temporal synchronization. The next issue is high dimensionality of shapes and also the shape sequences. The time length could be very large. And so we have PCA for shapes. So this is an example of an original sequence on the top and some sort of 5-dimensional reconstruction at the bottom. Towards the end, there's some sort of differences start emerging, which could be bigger, but 5 is rather low. One can use some higher dimension. One can also do the temporal time dimension reduction. So we treat these sequences as functions. Once you are in the tangent space, then these are just simply Euclidean functions, and functional PCA provides tool to reduce the time into smaller coefficients. And we have done that. You can do joint space and time PCA to reduce these very high dimensional sequences into low dimensional representation. So I'm going to show you 3 examples of applying these ideas. One was cell motility and amoeba histolica. And so we have videos of these things under different, in Petri dish under different conditions, and they were interested in introducing inhibitors to see if you can control the motility and so on. The kinematic side of it is completely removed. So in answer to your question, we even removed the scale. Here we simply focused on the shape evolution. We could have kept something if we wanted in the analysis, but we removed everything. Rotation, translation, and scale. And then after doing PCA, we got this sort of discrete time series. We fitted classical time series models. And so each sequence through this process is represented by a set of parameters that estimate the dynamics in the shape space. And then we compared the sequences by comparing their parameters. So one VAR model to one sequence, another VAR model, and we compared the model, and we got some sort of reasonable success in classifying whether an inhibitor was used in this dynamics or did it make a difference in the shape dynamics along that. This was the example of a person performing activities. On the left is an original sequence. It sort of looks rather simple, and think, why can't some deep neural network do this? The goal was to take such sequences on the left and generate simulated sequences on the right. But the issue is there are very long sequences, 1 minute, 2 minute long sequences, and very small data set originally, like 60 sequences, that's it. So using 60 sequences, so we have to learn. And of course, in statistics, when you have small data, you go to parametric models. So we developed this sort of pipeline again, synchronize the sequences, flatten them, do the spatial and temporal PCA, and impose Gaussian models on these reduced representations. And we were able to get, some kind of half decent simulations. That's where we are currently. There are some postures that turn out to be completely ridiculous individual postures, but we are in some high dimensional space, and we have not imposed any physical dynamics. We haven't introduced any biomechanical model on joints or the length of the arms, none of that. This is simply purely data driven. Give me data, I compute mean and covariance, and I generate new Gaussian random variables, right? Okay, this is the example that I'm quite excited about. This is relatively new, but it brings in a lot of challenges. So tomato plants, these are actual observations. Each row is growth of a tomato plant, growing from a seedling into a bigger plant with branching structures. And you have timestamps for these, and you are missing data. So for each row, you have some observations, and there is no notion of synchronization. I mean, somebody gave us data with timestamps, right, for each plant. Right? So we have to take the picture on the left and make it a picture on the right. Firstly, we have to fill in the missing. We have to decide what time frequency we want. We have to, through some sort of interpolation tools, geodesic tools, spline tools, fill in the missing shapes, and we have to register them. We have to reconcile them across observations, so vertically. And so we have done that. And so again, Vanessa's question, how subtle differences can still be registered? Toward the smaller side, they look quite similar, all of them. Still, there is some sort of registration in place. They have different number of branches. They are topologically different. And so all of this has to be inferred in a data-driven sense. And then we have this four-dimensional geodesic. So we have on the top row the registered growth of one plant, and the bottom there is a registered growth of another plant. And vertically we interpolate between them as individual shapes. So we can just interpolate between them and quantify how different these growths were for these different plants. In fact, we can compute an average growth model by averaging over hundreds of these plants, interpolated and registered, and then shapes averaged along after registration. We have not only means, we have sort of these covariance models in space and time, and we can generate some new growth models, completely simulate random trajectories in this space which look like this. And one can see sometimes it goes completely off, ridiculous and topologically incorrect things, because we didn't impose any other information. We are simply trying to do this from the data. So I think I'm going to stop here and leave time for some more questions. One of the important slides I always show, I don't know whether this is applicable to this audience or not, but geometry and statistics, that's a wonderful combination. You have a lot of data which has structure to it. The whole world has a lot of structure to it. And we observe it, we record it, and we want to be able to do statistics with it. Unfortunately, the traditional statisticians are ill-trained to deal with this sort of non-Euclidean, non-linear constraints that are imposed. But I think the field is emerging, and hopefully there will be many more people. One last pitch I want to make is that a lot of these similar data sets or similar problems may be available with some of you, and I'll be very happy to participate, collaborate, whatever we can do to help out. Even if there are some sub-problems like registration or interpolation or anything that I can do to help, I will be very happy to help. Because ultimately that's the goal, is to make some difference. I'm going to stop here.